and introduce some of the terms that may not be as familiar to all in this particular arena. My disclosures are listed in the AGA handbook. What I'm going to do is focus on several aspects of biosimilars, the economics, what are biosimilars, some of the EU and the FDA, if you would, definitions, and then to talk about the biosimilar landscape and a little bit about the technology directly. We're fortunate really to have terrific speakers today, which I'll introduce as they come to the podium. And one of the things that's recognizable is that in 2009, the Biologic Price Competition and Innovation Act came to place, and this is an abbreviated licensure pathway that is done to be showing biologic products help gain entry into market if they're biosimilar or interchangeable through an FDA licensed product. If we look at the overall economics of biosimilars, based on some data that was published in 2015, they accounted for 1% of all prescriptions, but accounted for 28% of drug spending. So these are some very important economics to keep in mind. If you look as well, the average cost in 2013 for a biologic agent in the United States per day was $45 versus $2 for a chemical drug. Global sales estimated back a while in 2013 to be, by 2017, were estimated to be $180 billion. If we look at this, about 50% of the sales coming from 11 biologic agents will face exclusivity loss by 2022. And the cumulative savings that have been estimated, and they vary, but these are some of the numbers, in the EU and the US as a result of biosimilars could exceed $56 billion in aggregate over the next 5 years, up to $112 billion. So these are things that can be advantageous to healthcare in general. The cost is considerably more than a small molecule, given the aspects of the manufacturing, the quality control, the marketing, the storage, the special requirements for pharmacovigilance, and they're not as able to be discounted as much as these small molecules. Small molecules may be 50 to 80% discounts compared to the originator products. In the EU, if we look, there had been about a 30% price reduction based on recent publications. And if you look across Europe, they can go as far as 69% for CTP13, which is in Norway directly. So they vary by region and they vary by government status. So you may say, what are biosimilars? And there's several definitions that have been put forth, but I use one that the FDA has used on their website. The biologic product is highly similar to the reference product, notwithstanding minor differences in clinically inactive components. And there's no clinically meaningful difference between the biologic purity and the reference product in terms of safety, purity, and potency of the product. So with that in mind, they have to be essentially the same strength, the dosage, injectable, for example, they have to be, and the route of administration needs to be the same as the originator directly. So you may say, well, how else do they differ? And these are some of the differences one might see. The composition, protein versus organic compound. The structure is a variable 3D structure compared to a well-defined structure. The route of administration, IV or sub-Q, and degradation differs, as well as mechanism of action. Blocking or depletion as opposed to enzyme inhibition is a characteristic prototypic examples directly. Manufacturing costs we recognize as well, and they require more labor and regulatory aspects when it comes to biologic products manufacturing, and it's a more expensive quality control and stability testing. So these are things that differentiate in general strokes, but not individually. And if we look at terms, you may see terms out there as a bio better or a bio superior. And that is not a biosimilar. This is a new class of biosimilars which go beyond the mimicking of the originator product, and they provide improvements, if you would, in one of the various aspects of the clinical profile through changes in the chemistry, the alteration in the formulation, and the innovative delivery, if you would. And we're going to talk some about interchangeability, so I'll define that term, if you would, the medicinal practice of changing one medicine for another that's expected to achieve the same clinical result in a given clinical setting, and in any patient that should be on the initiative or with the agreement of the prescriber. So let's look at some of the principles, if you would, that have been set forth, and these are extrapolation of indication, and I'll define what that is. So it's scientific justification for extrapolation should really address several issues. The mechanism of action in each condition in which the particular agent is searching licensure, the pharmacokinetics and the biodistribution of the product in different patient populations, the immunogenicity of the product in different patient populations, differences in expected toxicities in each condition and use in patient populations, and any factor that could affect the safety or efficacy of the product in each condition of use in patient population for which licensure is sought. Now, these differ from the EMA suggestions, and I put these up for completeness sake, but will not review these at this point in time. There's some fine lines of differences that the US and the EMA has directly. So to put this in a schematic, you may have examples as what we have are a phase 3 study in rheumatoid arthritis and a phase 1 study in ankylosing spondylitis that led to extrapolation of the indications of agents already approved. So this is, for example, infliximab, as we know it today in clinical practice, had been approved for Crohn's, ulcerative colitis, psoriatic arthritis, pediatric Crohn's, and plaque psoriasis, but not necessarily pediatric ulcerative colitis. And again, these are things that we can talk about. The majority were extrapolated based on the biosimilarity exercise by comparing it to the product we know with infliximab as Remicade. And we'll talk about the nomenclature, if you would, of the different agents. And it's really based on the totality of the evidence. One size does not fit all, and this was borrowed from the FDA website directly to show what was there. Clinical, animal studies, clinical immunogenicity, clinical knowledge that's post-marketing experience, pharmacokinetics, pharmacodynamics, and structural and functional characterization. So the one-size-fits-all hypothesis is not the situation that comes about, and the FDA scientists will evaluate the applicant's integration of various types of information to provide advice on scope and extent of the developed plan, and ultimately an overall assessment that's biologic product or is not biosimilar to an approved reference product. So that's the general concept. And if you look directly, different regulations exist in different countries. The US, the EU, Japan, Canada, WHO has suggested things, and they're similar, but they're not the same, if you would. So these are the biosimilar equivalents of the extrapolation. And when doing so, one looks at the totality of the evidence. The amino acid structure would be part of the quality of the structure and function. One would look at pharmacology and clinical as well. Differences in glycosylation, post-translation modification, and you should match the effector function regardless of the potential contribution to the mechanism. The pharmacokinetics in healthy volunteers is the most sensitive population in general to assess the kinetic similarity. Equivalent PK and pharmacodynamics with dose response experience can infer clinical efficacy if they're sensitive and the relative marker is available to look at. And then clinically, clinical efficacy in randomized, blinded, head-to-head study in a sensitive population with sensitive endpoints is advocated directly. Clinical safety confirmed in at least one sensitive patient population used as monotherapy with enough exposure and time to gain evidence from the data. And then immunogenicity with drug-tolerant assays in sensitive population with no concomitant immune suppression. And these in aggregate are looked at. And each step has a critical contribution to the totality of the evidence. that's being there, and each step should rely on the most sensitive state-of-the-art capabilities. No step can refute or overcome differences in other development steps, and it should satisfy all three steps to determine biosimilarity. So just as a background, what's been done. In the U.S., there were two trials looking at infliximab, CTP13, the biosimilar, and the trials were such that they were done in different disease states. Rheumatoid arthritis, and the Planetra was a phase 3 study, and ankylosing spondylitis, the Planetas study, was a phase 1 study. The population was RA versus ankylosing spondylitis, and the numbers of patients were significant, and various endpoints were looked at. The ACR20 at week 30 and the PK equivalents at steady state. And these were then evaluated and the endpoints were met for the studies. Now, this is of interest because there's now over 20 biosimilars in various phases of development in the US and abroad, looking at infliximab and adalimumab alone. And these are the agents that have come at the time where they're no longer exclusive, if you would. Different countries do different things. If you look in Germany, payers are giving targets for physicians prescribing of biosimilars. In Norway, payer attention to pharmacists to substitute has been something that has been done. Amgen went against this, they sued and they won, and Norway is now appealing. Not appealing, now appealing. And Brazil has separate things directly, so they differ throughout the world as to how things are done. We currently have a nomenclature that is accepted, and it's different from the standard biologic nomenclature. In August 2015, the FDA set forth a rule for naming biosimilars, and the name includes distinguishing suffixes which are devoid of meaning, and it's 4 random lowercase letters. So, for example, Remicade as we know it is infliximab HJMT directly. The intention is to avoid inaccurate perception of biosimilar efficacy, influence prescribing patterns directly. And then we have infliximab DYYB, or Inflectra, which gained approval April 2016, infliximab ABDA, which is Renflexis, which in April gained approval, and then adalimumab ATTO, otherwise known as Amjevita, which gained approval in September 2016. So this is easier for tracking if there is a safety issue, and also for distinguishing and saying that they are biosimilars. There are many questions that come to mind, I'm sure, in the audience and in people's direct consideration. We're going to answer some of those today for you. So I pose, the pathway to biosimilars is a work in progress, but a lot is known, a lot has been done. How comfortable will physicians be prescribing agents? How comfortable will patients be using biosimilars? How will insurers manage these agents? Can we switch from native biologic to biosimilar without immunizing the patient? And we'll talk about some of that. And can we do a double switch without immunizing from native to biosimilar and then back to another biosimilar? And these are questions that are obvious in the mind of clinical practice as well as investigators.